Yes.
Yes, you're audible.
Okay. Is my screen is visible?
Yes.
Okay. So as Dr. Vishwanathan clearly covered on the XFEP part, it is a free energy perturbation. Now I am going to show a practical demo on how to perform those calculations. Okay. So the platform which I am going to discuss today is XFEP. Okay. In order to perform the free energy perturbation calculation, you can use this platform, and this platform has been hosted in Amazon US cloud. Okay, so for example, you can just download the software from this platform and you can install in any of your laptop or desktop, and you can perform the calculations. The hardware will be used from the cloud. Okay, so now let me show you how to perform the calculation. So in order to perform this free energy perturbation, you need a protein structure information and docked ligand information to perform the simulation and get the value. Okay. So how to get started here is, once you log in into this platform, you can see this interface. Okay. In this interface, first what you have to do is you have to create a project. Okay. You have to create a project. Once the project has been created, then it will be good to go. So now I will show you the demo on the project which I have created previously. Okay. So first we need to prepare the protein. So how to prepare the protein is, just click on the submit option. The submit option, there is an option called protein preparation. Okay. Just click on the protein preparation. Once you click on the protein preparation, then you can add your protein. Okay. Click on add protein. So here what you can do is, here there are two options. One, you can just type your PDB ID and you can get the protein which is directly downloaded from the RCSP protein data bank, or else you can also upload the protein structure from your local directory like this. Okay. Then once you uploaded the protein, you can see the protein in the 3D interface here. Then there is an option to delete the residues. For example, you want to delete the waters, or if you want to delete the co-crystallized ligand, or if you want to delete some amino acid, those things can be deleted here. Just you have to click on it. Accordingly, it will be deleted during the protein preparation. Then, if you want to model the loop, if you want to fill the missing loop, that also can be done in this platform. So currently this platform supports loop modeling up to 10 amino acid residues. Okay. Here you can see the sequence of the amino acids here. You can identify the missing loops and you can fill the missing loops. Currently, you can focus on filling the loops which are in the binding site. Binding site is nothing but, for example, if you have a crocus ligand, the surrounding sphere of 6 angstroms are considered as your binding site or active site. Okay, so you have to concentrate modeling those missing parts. Okay, then you can define the protonation state, at what pH you want to prepare the protein. Then if you want to cap the residues like C terminal or N terminal, that can be done here. And if you want to add the hydrogen atoms for the ligand and cofactors, that also can be done here. After putting these parameters, just click on the run. Okay. Once you click on the run, it will automatically prepare the protein. Then the prepared protein will be available here. So once the protein has been prepared, here you can see it will show you the 4-letter code of the PDB and which chain you have retained, whether it is prepared or not. If it is prepared means it will be shown as prepared. Then if you want to see the 3D structure of the prepared protein, how it looks, you can see it here. If you click, you can see the prepared protein here. Then if you want to modify any of the residues, or you want to titratable, if you want to modify any titratable residues, that also can be done here. Okay. And you can, okay. Then in case of free energy perturbation, there are two different methods. One is RBFEP. What is RBFEP is relative binding free energy calculations. Another one is absolute ABFEP. ABFEP is nothing but absolute binding free energy calculation. So when you should use RBFEP is, for example, you have designed 20 ligands. Okay, those 20 ligands belonging to the congeneric series. Okay, mean to say only a small changes in the substitutions. If you want to study what would be the binding affinity of those compounds, in this scenario, you have to use the relative binding free energy calculations. This is number one. Scenario number two is, for example, you have designed 10 compounds. Those 10 compounds are novel, which don't resemble each other. Those 10 molecules are unique. In this scenario, you have to use ABFEP, which is known as a absolute binding free energy calculations. Okay. So now I am going to show you how to perform RBFEP. Now I have prepared the protein, right? Then I have to pair and map the ligands in case of the RBFEP. How to do the pairing and mapping here is, it is very simple. Just click on the submit. In the submit, there is an option for pairing and mapping. So this FEP can be done for small molecules, macrocyclics, peptides, GPCRs, PROTACs, as well as for covalently bound molecules. All those type of compounds can be studied by using this method. And also, if you want, if for example you want to understand the changes in the binding affinity of the compound due to the mutations in the protein, those type of things also can be studied in this platform, which is known as a protein mutation task. Okay, so now I am going to take the topic of RBFEP and show to you. Okay, now I have prepared the protein. Then I have to do the pairing and mapping of the ligands in order to perform the calculations. Okay. So how to do the pairing here is, just click on the submit, then click on the normal ligands. Okay. If you click on the normal ligands, it will open a panel like this. Okay. So here, what you have to do is, so this will be the window. Here, the input for pairing and mapping is the docked 3D confirmation of the ligands has to be uploaded here. Here you can see now I have uploaded almost 31 ligands. Each and every file contains single confirmation, single ligand confirmation. Okay. Here I have to upload the ligands from the local directory. Then here there are three type of pairing. One is radial, next one is low map, third one is opting. So depending upon your data set, you can choose this method. Then another option here is hub molecule. What is hub molecule is? So whatever the molecule you are choosing as a hub molecule, that will be taken as a reference, and it will be showing you what is the Tanimoto similarity between the reference compound and the designed compounds. Okay, so here you can see I have chosen the compound number 13 as a reference. Now it has taken the molecule 13 as a reference, and it is showing you for each and every ligand what is the similarity. Okay, then here you have to click on the generate expiry. Okay, so this will generally take some time. It will take around 5 to 10 minutes of time in order to complete this task. Once this task has been completed, you can see the output like this. Here you can see the compound number 13 has been taken as a reference, and whatever the molecule which I have designed are mapped And for each and every ligand, it gives you the similarity. What is the Tanimoto similarity between the reference molecule and the ligand structure? If you want to see the mapping in 3D, that also can be done here. For each and every compounds, you can see the mapping in 3D. So whatever the color which is in white color are the substitutions apart from the reference molecule. Okay. So once this pairing has been completed, then directly you can submit the compounds for performing the calculation. Here you can see this is the panel where you can see the pairing and mapping results. Then here there is an option to submit the task. Okay. Once you click on submit the task, it will directly take you to the panel. Okay. So here what you have to do is here you have to give the input, name of the compound or name of the study. Then here you have to upload the prepared protein. Okay. Since we have already prepared the protein, you have to choose what is the protein you have prepared. Then here it will automatically load the output from the pairing and mapping task. And in the bottom, you can see the ligands, the protein structure and the ligands, which are perfectly aligned and are good to go for performing the calculations. Then, in case of RBFEP, you have to perform the round of calculation thrice. Why? Because the value will be, it will perform the calculation thrice and the values will be averaged out, which will give you a more efficient results. Okay. Then here there is something known as a production time. This production time is nothing but simulation time. So the production time stands ranging from 1 nanoseconds to 50 nanoseconds. For example, if you are performing a production run of 5 nanoseconds, 1 nanoseconds will be considered for equilibration. Remaining 4 nanoseconds is for the perturbation. Okay. Likewise, you can go up to 50 nanoseconds for simulating and equilibrating and doing the production run. Then here, in case of performing the free energy perturbation, there is something known as a lambda. So if the difference is bigger with respect to each and every structure, you can increase the lambda windows. Generally, we give you the option of 3 lambda windows, which is 12, 16 and 24. So you can increase or decrease this lambda window depending upon your structure of the compounds, whatever you have designed. Okay. Then here you can click on run task. If you click on run task, this task will be automatically submitted in our Amazon US cloud and you can see the status of the calculation in the task monitor. In the task monitor, you can see what is the status of the here you can see in the task monitor what is the status of the study. Okay. Then once the calculation has been initiated, that also can be seen in the job monitor and the output will be looking like this. Okay. So here after pairing and mapping, if you submit the calculation, the calculations will be initiated and you can see the output here. If you want to see the output of individual calculation for each and every pair, that also can be seen in the here. For example, 13 is the reference compound and 14 is the design. If you want to see the convergent analysis, you can see here. Likewise, if you want to see the trajectory, what is the RMSF, what is the RMSD deviation, and what is the H-bond interaction which has been maintained, what is the partial charge, what is the dihedral angle, all the things can be seen here, and what is the pairwise energy, all the things can be seen individually, and you can also download those reports in the form of a PDF file. Here you can download the output as a PDF file. Then for each and every task, whatever it is performing, it will give you the delta delta G, which can be directly correlated with the experimental activity. Then also it gives you the uncertainty value, the standard deviation, and the average uncertainty. If you want to see the map of the perturbated thing with the standard deviation, that can be seen here. Then, for example, if you have your IC50 data, you can input your IC50 data and you can directly correlate with the predicted results versus experimental results. And also, it also shows you what is the graph between delta delta G experimental versus delta delta G predicted by FEP. Also, it shows you the difference. It is another graph for delta G experimental and delta G FEP. It is automated, and if you want to download this graph, that also can be downloaded. So this workflow is for performing the RBFEP. This is number one. Likewise, you can also perform for ABFEP. In case of ABFEP, there is no need to do the pairing and mapping, because whatever the ligands you are going to perturbate is novel, and don't have the structural similarity between each other. So in that case, first you have to do the protein preparation. Then you can directly come to the submit ABFEP task. Okay. In the submit ABFEP task, you can generally upload the docked 3D confirmation, and you can upload the prepared protein. Remaining all the things are same, and you can submit the task. Okay. The pairing and mapping is required in case of RBFEP, whereas in case of ABFEP, it is not required. And we also totally have 5 different type of force fields, okay, which are proprietary only to us. One is XFF3 rest to force field. Another one is XFF3 rest to force field. Another one is specially made for peptides. These two are commonly known, which is EAM1 BCC and GAF2 force field. Okay, so this is a thing for this is a demonstration of how to do the calculation by using RBFEP and ABFEP. Then I will also quickly show you, for example, if you want to study the mutations in the protein, how the mutations would affect the binding affinity of the compound. That also can be studied here. So here you can see there is an option to submit the protein mutation FEP. So just click on the submit protein mutation FEP. Then here you have to upload the prepared protein. Once you upload the prepared protein, here you can add what are the mutations which are available in the protein. For example, which amino acid is mutated to which amino acid. Likewise, you can add the different mutations, whatever you have to perform the study. You can add up to 10 mutations you can add. This is option number 1. Number 2, for example, you have a protein, different set of protein with different set of mutations in the form of a PDB. That also can be uploaded in as a PDB file here. In the complex state, if you have the different mutations which are available, that also can be uploaded here in the protein data pack format, and you can upload the ligands. Then you can perform the perturbation. Okay, so this is regarding the protein mutation task. The another next option is, for example, if you want to perform the things for GPCRs. In case of GPCRs, you have to create a membrane before performing the calculation. So that option is also available here. Here, while preparing the protein itself, you have to select the membrane. You have to select the membrane. Then you have to give the orientation. Then you can prepare the protein. Then it is simple. So with this, I'm completing the demo part. So if you have any questions or doubts, you can unmute yourself and you can ask me. Hello.
Nicely demonstrated, Mr. Anbu. And that was an excellent presentation by Dr. Viswanathan. If anybody here has questions for either Dr. Viswanathan or Mr. Anbu, now would be the time to drop them in the chat before we quickly move to the next session. I see a few questions. So the first one is, what is the optimal range of time frame in terms of nanoseconds while running simulation?
5 nanoseconds.
Okay. What would be the selection criteria for off-reference molecule while doing Tanimoto index-based similarity search?
Okay. That's a very good question. When choosing the reference molecule, it is more preferable. Mean to say, whatever the reference you are choosing, it should have a experimental activity which is reported. You can choose that as a reference and you can proceed for the calculation. And there is no similarity cutoff as per my experience. So the similarity cutoff will be ranging from you can choose the compounds up to 0.30 similarity. The lowest would be 0.30 Tanimoto similarity index.
And the more important thing here is, if in the Excel structure you have a particular molecule bound, that really helps to keep that as a reference molecule as much as possible. You can also use some other molecule, but the Excel structure advantage is that its binding mode is precise.
Any other questions?
And there is another question during protein preparation, is it necessary to remove water molecules and other associated ligand molecules? Actually, it is important to keep some cofactors or other structures if you have parameters for it, because that is how the Excel structure is obtained, and the atomic locations have to be correct. So that's possible only when you have the associated ligands. And also for waters, if you have water molecules in the hydrophobic pocket, they can be easily removed. If there are polar pockets where water molecule is present, which is engaged in interaction, you either keep it or you replace it with a group on the ligand, so that you capture the same interaction. So you have to study each case separately. Any other question?
I think we are good on all the questions. Last call for anybody who might have a question, then we are moving to the final session for today. We'll give it 1 minute.